My lab works with one of the molecule, which is called molecule of life, which is RNA, because everything pretty much revolves around this molecule. All the living organisms use this molecule heavily. RNA is a very dynamic molecule, and RNA and the amount of RNA that we produce, or our cells produce, define who we are. Understanding how it operates in cells makes it easier for us to piggyback on natural processes and fix some of them which malfunction.
We work with nucleic acid nanoparticles, or NAMs. And according to the sequence that you use, you can generate nanoparticles that will fold into different conformations.
Using NAMs for these particular applications was a completely new, novel direction that they took. And it has never been done before, and that's why it was not a trivial task.
And during bacterial infection, oftentimes we think of inflammation as always a positive thing. However, it can also be really damaging. This is really exciting because the aspect of nanotechnology is really great because it can have these immunomodulatory functions where we can tailor the immune response.
And if they fold correctly, they perform a certain function which is essential for us. And when we extract them, it becomes like a Lego block. We can build a certain architecture with a certain geometry, a certain size, and dimensionality and so forth.
That's where the AI ties in. We've taken all that information, and using AI, we can generate ways that we can predict outcomes from novel sequences, novel structures.
Based on the findings, we created a program which is called AI cell, artificial immune cell, which is now publicly available for everyone, where anybody can go to, plug in sequences of any NAMs. Within the 2 seconds, they will get a predicted output. So instead of spending 24 hours or 48 hours in the lab, you can do it within 2-second time.
It allows us to take this large amount of information that we've gathered and use it to generate means to predict an outcome.
By doing this, we can eliminate lots of side effects associated with the one-for-all treatments. We can reduce the dosage, the duration of procedure, and so forth, and make it more efficient for the patient eventually. That's the ultimate goal.
It's really exciting because we've done a lot of this initial characterization of this technology, and the more we characterize it, the more it can be applied to, again, a variety of infections and diseases. It would be very challenging without the use of AI. It should streamline selection of therapeutics for a given patient, and it should allow them to be more tailored to that patient as well. We're not there yet, and there's more to do. Which, that's the exciting part, but that's the ultimate goal.